Okay, good morning, everyone. So thank you for joining this lecture on quantitative proteomics and omics data analysis. So I'm Holger Kramer. I'm the head of mass spectrometry at the MRC Laboratory of Molecular Biology. And this is really an introductory lecture to methods used for quantitative proteomics data analysis, and then a closer look at how this data can be processed and visualized towards the end of the lecture. And just to give you an outline of what I'll try to cover here. So I'll compare a few different approaches to quantitative proteomics. So initially looking at methods involving stable isotopes, such as SILAC and the Isobaric labeling methods, iTRAQ and TMT. And then I'll compare that to label-free quantification. And then going on from there, I'll compare a few different approaches to really acquiring data in the mass spectrometer. So this will be targeted proteomics in contrast to two different types of acquisition methods called data-dependent acquisition or DDA and data-independent acquisition DIA. Then I'll exemplify the data analysis a little bit more in looking at the label-free quantification workflow in MaxQuant. And then I'll use an example of a fairly simple experimental design to show how then process data can be visualized and a statistical data analysis performed in a package called Perseus. So just sort of two introductory slides that are a brief recap. And essentially what I'm showing here is that peptides, polypeptides in the gas phase in a mass spectrometer can be ionized and then fragmented. And the fragments that are produced by this approach, they have this nomenclature here. And the most important ones to remember really are the ones that are generated by breaking polypeptide at a given peptide bond, basically between the carbonyl group and the nitrogen of the amide. And that produces a C-terminal fragment called the Y ion and an N-terminal fragment called the B ion. And those are shown here. Now, the B ions, they can undergo further degradation and lose a molecule of carbon monoxide, and then they convert it into an A ion. And the C-terminal fragment here, the Y ion, is shown on the right-hand side. And essentially, any data at the intact level before fragmentation, this is called MS1 level data, or also survey scan data. And any data that is produced through this process of fragmentation is called MS2 level data or fragmentation level data. And here's an example of this process. So where a sample has been digested with trypsin, analyzed on a tandem mass spectrometer, and then a fragmentation spectrum was obtained. And computationally, it is possible to now assign a peptide sequence, an amino acid sequence, to a spectrum of this sort. And this is shown here in an algorithm called MASCOT. And in MASCOT, we see that this matching between theoretical fragment ions that could be calculated just based on the amino acid sequence, how they can be matched with experimental peaks, so experimental fragment ion masses. And this is shown here by highlighting all of the matched fragments in red. And we observe that there's a lot more Y ions as opposed to B ions. And that's actually quite a common thing to observe with tryptic peptides. Now, there's one more important point. So we just sort of introduced that what you have to do through bioinformatics approaches is to compare the experimental spectra produced through fragmentation of peptides to theoretical expected spectra, which you can generate by taking all of the human proteins, predicting in silico the tryptic peptides from all of these proteins, and then calculating what the B and the Y ions would be expected to be, and then matching these, and then having a robust algorithm that will look at the likelihood of something being a non-random match. Now, one important concept as well that's being used in this approach is to generate false discovery rate, or FDR. And the way that's commonly performed is to essentially take the same sequences from the protein sequence database, so also called the target database, and then try to generate a set of random sequences. And the way that's usually done is by inverting the protein sequences and generating triplet peptides from these inverted sequences. So the idea there is that this should generate a so-called decoy database, which has the same overall size and amino acid composition as the target database that we're going to search or match our data against, but one that now only contains sequences that we expect not to be present in the sample. And then any match between a spectrum and decoy database is a means essentially of quantifying the extent of false matching or false positives. Now moving on to specific strategies used for quantification in proteomics. So quantification really is the concept that we don't just want to identify the proteins that are present in a sample, but we want to be able to do quantitative comparisons across a set of samples. And one of the early approaches that was quite successful is a method called SILAC, or stable isotope labeling by amino acids in cell culture. And the concept is shown in this diagram here. So the idea is that we could have cells that grow in culture, and that we want to label with different versions of amino acids. And the versions that are available to us are either the standard light amino acids, such as lysine and arginine here, or versions thereof which have a specified number of heavy isotopes incorporated in them. So here's an example where there's 4 deuterium atoms incorporated into lysine, or here an example where there's 6 carbon-13 heavy carbon isotopes incorporated into arginine. And that can be pushed even further, the concept. So by replacing all of the carbon and all of the nitrogens by their respective heavy versions, carbon-13 and N15, we can then have an 8-fold labeled lysine derivative and a 10-fold labeled arginine derivative. Chemically, these stable isotopes, they behave exactly the same as their light versions, but it can be distinguished in the mass spectrometer. So the important point now is that we need to grow the cells in media containing these other amino acids, and we probably need to propagate the cells long enough, 5 to 10 cell doublings, to really replace all of the, or to give the cell population a chance to expand, and to replace all of the amino acids present by their heavy, medium or heavy versions. And it's possible to check this, so to collect a small sample of the cell population, analyze it by Tandem Mass Spectrometry, and check the degree of labeling. And then once we are satisfied that the labeling is sufficiently complete, let's say 95% or greater, then we can perform an actual experiment. So this could be a wild-type control population of cells, then a set of cells where we overexpress a gene, and then another set of cells where we perform knockdown of that same gene. And then once we've performed that and have concluded that biological experiment, we can wash the cells, harvest them, and actually pool them, and essentially perform an experimental workflow for sample processing that will include denaturation of proteins, reduction and alkylation of cysteine residues, and trypsin digestion. And we can do this with this pooled sample of light, medium, and heavy conditions. And that has an advantage. It means that any comparison and any variability in sample preparation now affects all of the different conditions in the same way. So we're not introducing any more variation into the experiment. And now there's one particular advantage of using lysine and arginine in this setup, which is that trypsin is a protease which cuts C-terminal to lysine and arginine. So we expect that every triplet peptide should contain at least 1 lysine or 1 arginine residue, with the exception of the very protein C-terminus. And in the experimental design that's shown here, we would now expect that we distinguish these 3 conditions: light, medium, and heavy. And they have a mass shift of 4 Daltons for any of the lysine peptides, and a mass shift of either 4 or 6 Daltons, depending on whether we compare light to medium or medium to heavy for the arginine-terminated peptides. And that's intrinsically powerful, this workflow, simply because it means that we can actually do the quantitative comparison within the same mass spectrum, because any of these peptides with the different medium or heavy labeled versions, they will behave chemically the same, meaning in any separation, like liquid chromatography, they will elute off the chromatography column at the same retention time. Okay, so then just to give an example of how this kind of workflow can be applied. So what is shown here is actually a strategy to utilize SILAC to look at the identification of enzyme substrates of an enzyme that's called factor inhibiting HIF. So factor inhibiting HIF is a 2-oxyglutarate dependent dioxygenase, which hydroxylates asparagine residues in substrate proteins. And the reaction scheme is shown here, where the unmodified amino acid residue has got a carbon-hydrogen bond, and the enzyme utilizes dioxygen from the atmosphere and iron 2 as a cofactor to now introduce a hydroxyl group to hydroxylate the amino acids, and at the same time convert 2-oxyglutarate to succinate and carbon dioxide. Now, there is an enzyme inhibitor available for the whole group of 2-oxyglutarate dependent dioxygenases, and this one is N-oxalylglycine, or the cell-permeable version thereof, which is a dimethyl ester thereof, which is called DMOG. And the whole concept here now is to say, okay, substrate proteins are substrates. They're not actually binding partners of FIH. So therefore, we expect that any binding events would only be transient as part of the enzyme transition state. So if we are trying to now essentially freeze these in time or be able to assay these, we really need to stop the turnover of this reaction by adding this inhibitor. And the idea is then, if we're then able to perform in immunoprecipitation in the presence of this inhibitor, we may be able to assay candidate substrate proteins in that way. So that was performed here. So basically here it's a simpler design with just the light and the heavy condition. And in both cases, expression of FIH is induced by a doxycycline. But in the heavy condition, in addition to the doxycycline induction of FIH expression, we also add DMOG, so the cell-permeable enzyme inhibitor. Then we perform an IP, an immunoprecipitation with a FLAG epitope. And then we pull the elutions from the IPs. And then in the pooled sample, we perform an in-solution digest and then liquid chromatography tandem mass spectrometry analysis. And the outcome of that experiment is shown here, where we now have a set of proteins which are enriched in the heavy condition, and which are only seen at much lower levels in the light condition. So any of these are now candidate protein substrates of FIH. And then the important step to really confirm whether these indeed are substrates was to perform another experiment overexpressing, comparing FIH overexpression to endogenous FIH levels or to knockdown of FIH, and then really observing the levels of the modified peptide, in this case, in this pancrease 2 substrate. And indeed, what we see here with FIH overexpression, we get an increased level of that modification, 70%. At endogenous levels, it's much lower at only 7%. And with FIH knockdown, we only see trace levels of less than 1%. So that essentially this experiment then confirms that indeed pancrease 2 is a genuine substrate of FIH, and the SILAC strategy allowed us to identify that. So now I would like to compare this approach to a different approach, which has been termed isobaric labeling. There's sort of two different versions of this approach that were commercialized. The first one was called iTRAQ, and I'll start on that one, and then I'll move on to the TMT reagents. And just to explain the concept, so this was introduced by Darrell Papin, who at the time led a proteomics laboratory at Hammersmith Hospital campus, and then also worked for SciEx. And essentially the idea of this concept was the following: to create a reagent which can react with peptides through this active ester. So these active esters will react with amines, either on the N-terminus of a peptide or also on a lysine side chain. But the most important point is you can attach this group to the N-terminus of any peptide that has a free N-terminus. And then what we have here is a different distribution of heavy isotopes between different parts of this reagent that's then attached to the peptide. some of these heavy isotopes can be incorporated into this ring structure here as carbon-13 isotopes. Or alternatively, they can also be incorporated into the carbonyl group itself by either labeling the carbon as carbon-13 or labeling the oxygen as oxygen-18, or a combination thereof. And that essentially allows us to distribute heavy isotopes either between this group here, which is called the reporter ion, or this balancer group, while leaving the overall mass of the reagent the same. And so what does that do when we then analyze this in the mass spectrometer? Essentially, what that leads to is that the bond between the carbonyl group and this ring structure here is broken, and we liberate these reporter ions. That means under MSMS fragmentation conditions, we then are able to see 4 peaks at 114, 115, 116, and 117 Daltons. And that is shown over here. And this was really a test experiment to demonstrate the concept where a mixture of proteins was labeled with the 4 different reagents with the 114, 115, 116, 117 reporter ions, and was then combined in a 1-to-1-to-1 mixture. And in the low MOZ region of the fragmentation spectrum, this is what observed. And then importantly, if you make a different spike in mixture where the ratio differs, so where we have a 1-to-5-to-2-to-10 mixture of this protein mix there, then that is indeed what is reflected in this lower MOZ region in the MSMS spectrum of in theory, of any peptide from these proteins that have been labeled in that way. Now, importantly, it's then important to quantify how accurate this actually is. So the mean ratios that were obtained across a whole set of different peptides were determined, and the standard deviations were determined, and showed that this can be a good approach to look at different quantities of these peptides at the MS2 fragmentation level. How could that now be used for quantitative proteomics experiments? And this is shown here. So if we imagine we have a set of 8 samples, and it's important to highlight that there is another set of these reagents that were created and synthesized, which allowed 8 different channels, so 8 different reporter ions to be liberated under MSMS fragmentation conditions. So the idea then is we have the set of 8 samples, all of which are processed and digested separately. Then each of these 8 different samples are labeled separately with one of the flavors of this labeling reagent. And now the reporter ions go from 113 to 121. Once all of these samples have been labeled separately, it is now possible to pool the samples and then perform instrumental analysis. And the instrumental analysis involves both data generation at MS1 level. So this corresponds to intact peptides. So one peak here we would expect to correspond to a single peptide. And when this set peptide is now fragmented in order to obtain the amino acid sequence of that peptide, we also observe in the lower MOZ region this fairly dense region here where there's a whole set of peaks. And these peaks now correspond to the liberated reporter ions that originate from the chemical reagent that we introduced to the peptide N-termini here. And the idea now is that while each of these reporter ions behave chemically the same and ionize in the same manner in the mass spectrometer, their intensities now should reflect the respective quantities of each of this peptide or the corresponding protein in the respective samples here. So that's quite an elegant approach. So it's in contrast to SILAC, where all of the quantification is done at MS1 level. Here, all of the quantification is performed at MS2 level. And just to highlight that of course, we have a limitation there. So the number of different samples we can compare is now dependent on how many different reporter ions we can generate from this type of reagent. And this over time did indeed become somewhat limiting. So over time, new types of reagents have been introduced. And what we're looking at here is a relatively recent iteration of this. So these are now the Tandem Mass Tag reagents, or TMT reagents. And they have a different chemical structure, but the principle is the same, that there's a reporter ion that can be liberated, that there is a different distribution of heavy isotopes between the reporter group and the balancer group. And that allows us to now multiplex this across a set of samples. One further concept that then was introduced was the fact that in a high resolution mass spectrometer, it is actually possible to distinguish the small mass difference that originates from either heavy nitrogen or heavy carbon isotopes. And that is shown here. So in the bottom panel, it looks like we're looking at 9 different channels from 126 to 134. But when we zoom into the peaks in the center here, we see that each of those come actually as a pair. And even though 127N and 127C both have a nominal mass of 127, at high mass resolution, they can be split, where the heavier is 127.1310, corresponds to a heavy carbon 13, and the slightly lower mass of 127.1247 corresponds to labeling with heavy N15. So that essentially now allows us to increase the multiplexing capability of the approach. So in this example, 16-plex labeling of 16 different samples that can now be analyzed in the same LCMS experiment. And then most recently, we had introduction of 2 more flavors of this reagent to push this to an 18-plex experiment. And the mass difference of 6.32 millidaltons between heavy nitrogen and heavy carbon means that we need high resolution at MS2 level, so at the level of the fragmentation spectra, to be able to distinguish this. So fortunately, that can be done, for example, in Orbitrap-based mass spectrometers. Just to highlight maybe a few features of this approach. So there can be challenges to performing isobaric labeling quantification. And one of these challenges is shown here. So when you have a larger set of samples where 16 to 18 different samples might still be insufficient for the experiment to be carried out, you may be tempted to split your samples across different batches, different 16 or 18 plex batches, to get to the higher number of samples that you wish to analyze. Now, one of the big advantages of isobaric labeling has been the fact that these data sets have a very low incidence of missing values. So a missing value is if you've got a protein which was quantified in some conditions, but which was not quantified successfully in some other conditions. And that is a problem because it means that in some conditions, we know that there would be a low level of the protein, but we can't actually specify how low that level is. It's not detected. It's below the limit of detection. Generally, TMT-based quantification has a very low incidence of that, to the point actually where TMT-based proteomics is not very good at seeing the absence of a protein. So even if a protein is generally not present in a particular sample or a particular condition, it will probably be detected at a low level. But we'll come back to that later. So when we now split an experiment across a number of different TMT batches, we suddenly see that when we compare the different batches to each other, that now these missing values do start to creep back in. And when we look at the peptide level, we see that extent of missingness in the data can actually increase to up to 50% of all of the peptides quantified across a large number of batches. And this example shown here, that was where TMT 10-plex experiments were carried out to cover a very large set of samples. Now, the situation is not quite as bad at the protein level. So it's worthwhile keeping in mind that all of the proteins that are quantified, they may generate a whole number of peptides, strategic peptides that map to a particular protein. So when we quantify at the protein level, the situation is not as stark. So actually missing protein level intensities, they are lower, but still noticeable when we now combine a number of sets of different TMT batches. And then a final point to sort of doing these multi-batch TMT experiments, it's worthwhile actually incorporating one reference channel. So to use at least one of the 16 or 18 plexes that are available for essentially one sample that is run in all of the batches. And that can be used for normalization to compare the different plexes to each other, but then allows us as well to compare the data in this reference channel to the data quantified in each of the individual batches. And what is shown here is that the intra-batch variation of protein intensities, or here protein copy numbers that were quantified, within a single batch, they tend to be very low. But when we look at the reference channel and the quantification of protein copy numbers across all of the different plexes in this multi-batch experiment, then the inter-batch variation is significantly higher than that. So essentially, this is just to demonstrate that there are challenges when going beyond the number of channels available within a single n-plex experiment. Okay, so now comparing both of these labeled approaches to label-free quantification in proteomics. So label-free quantification in proteomics conceptually is actually very simple. The fundamental idea there is that we perform LC-MS analysis, where we have liquid chromatography, tandem mass spectrometry data. And from this data, we can extract traces where each of these lines here may correspond to the intensity of certain peptides present in the sample. And here we see one trace in red and one trace in green. And the idea is now that for certain signals, certain peptides, we may have an increased level and increased intensity in the red sample, or in others an increased intensity in the green sample. Now, even though that sounds conceptually very simple, it of course now means that each of the samples has to be injected and analyzed, run on the liquid chromatography system separately. So essentially, any variation that we have in instrument conditions between the different LC-MS runs will now affect the accuracy of quantification. So that is one challenge. And another challenge is that the stability of these systems may not be perfect. So when I run a system and do a liquid chromatography run that may take me an hour to analyze one complex sample, then when I perform the next injection and perform another run that takes an hour, the conditions may have slightly shifted, and that could lead to variations in retention time, for example, where certain peptides elute a little bit earlier or later. Now, there is methods to now improve on that, to essentially recalibrate these retention times and align the different chromatographic runs. And that is indeed performed. And then on top of that, it is important to actually define each of these signals and make sure that we only compare the same peptide in one sample to the chemically identical same peptide in the next sample. And the way to ensure that is that we determine accurate masses. So we need high mass resolution and high mass accuracy for these peptides. And after retention time alignment, they should also have the same retention time within a very narrow window. And a different way of visualizing this type of data is in this two-dimensional LC-MS chromatogram. On this plot here, what we have is the M over Z, or mass to charge ratio, on the x-axis. So that's what we usually expect as the x-axis on a mass spectrum. But now on the y-axis, we're showing retention time. So we're able, in this way, to summarize a whole LC-MS chromatogram of a run that may take 2 hours in a single map. And we can now zoom into a particular region of this. And what we're able to see here is at high enough mass resolution, we actually see the isotopic resolution of the peaks, and we see how they start to elute off the column, gain in intensity, and then decrease in intensity again. And essentially what we now need to do in order to process this type of data is to overlay different 2D LC-MS chromatograms from different runs and make sure that all of these runs are well aligned. Now, a little bit later, we'll come back to the label-free quantification workflow in MaxQuant, and we'll go into this type of quantification approach in a bit more detail. So now I would like to just move on to pointing out that there's actually different mass spectrometry acquisition strategies that can be used in quantitative proteomics. And the first one of these is targeted proteomics analysis. And essentially what targeted proteomics analysis exploits is the fact that when we perform fragmentation of peptide ions in the gas phase, we minimally know, if for a given peptide, we can predict the precursor ion mass if we know the sequence of the particular peptide of interest. And we can also predict the fragmentation pattern based on the known rules of fragmentation. And we looked at the B and Y ions that we can predict for these. And what we are able to do actually is we can define a peptide signal through both the mass of the precursor ion and the masses of various fragment ions. And that is something that can be exploited in a targeted proteomics approach. So what we're doing here now is not actually discovering what proteins are present in the sample. We're actually turning this whole method on its head. We're saying, okay, we have a set of known proteins that we're already interested in to compare across a set of samples. And what we would like to achieve is to quantify these known peptides with very high sensitivity. And what we really want to avoid is any non-detections, any missing data in the data set. And that's shown here. So essentially what we're looking for is combinations of precursor ion masses and product ion masses. And this transition from precursor to product ion mass through the fragmentation process, that's also known as a mass transition. In this example here, what we're looking at here is the combination of 4 transitions of the precursor ion, the intact peptide mass, to 4 different fragment ion masses. So in this case, the Y9, Y8, Y7, and Y6 ions. And we're able to then plot that as a function of retention time. And what we expect for any of these mass transitions that generally belong to a given peptide, that they should all cluster around the peptide retention time. So essentially the characteristic retention time at which the peptide elutes from the chromatography column. By monitoring this, we're able to now achieve a gain in sensitivity, because what we're now in this targeted proteomics approach are doing, when we're not trying to scan the mass spectrometer for any peptides that are present, but the mass spectrometer only spends time on acquisition of these mass transitions. So it's basically has got a very blinkered view of only looking at what we're interested in, what we already put into the experiment as the peptides that we want to quantify across a set of samples. And we can perform this by selecting these specific mass transitions. And the approach has been termed multiple reaction monitoring, or MRM. And then in the end, we'll use specialized software such as the Skyline software from the McCoss lab to extract all of this data and quantify across the whole set of samples. Now, because this is now not a discovery proteomics approach, this has actually been proposed as a follow-up experiment to discovery proteomics experiments, where we may have a large set of samples that we need to analyze, and where we want to be able to quantify without any missing values across the entire set with maximal sensitivity. And here's shown an example of targeted analysis of, in this case, actually a small molecule. And essentially what that shows is that when we have a dilution of a set of standards here of this particular molecule and in a highly specific transition, we can build a calibration curve. And on that calibration curve, we can identify a number of things, such as the linear range of quantification, the limit of quantification, and we can demonstrate that we're now able to detect this particular analyte with increased sensitivity. Now, when we apply this approach to proteomics, we need to be sure that the transitions that we monitor are actually specific to those peptides only. And there are bioinformatics tools to do this, to define so-called proteotypic peptides, where we bioinformatically ensure that these mass transitions are unique within the proteome of interest, so that we're not going to be plagued by contaminating peptides or peptides that happen to have the same mass and similar fragment ions. Now, in terms of the disadvantage of this approach, is we need standard peptides to be able to build an assay like this. So we actually need to order the corresponding synthetic standard peptides, and it is advisable to optimize the acquisition parameters so that we have maximal sensitivity for this approach. And then on top of that, we also want labeled standard peptides we can spike into the sample to take into account any changes in sensitivity due to other components that may be present in the sample. So moving on from this targeted approach to a discovery proteomics approach, and we'll first look at data-dependent acquisition, or DDA, and that is still the commonly used approach for most discovery proteomics experiments these days. So here we use the instrument in the following way. So what is shown here is an example of a Q-exactive Orbitrap-based mass spectrometer. What we use is we have the electrospray ionization interface, where we take the peptides into the gas phase and ionize them. We are then able to select specific peaks in the in the quadrupole. So this is precursor ion selection. And then we use this HCD cell for fragmentation. So this is where the MSMS fragmentation actually happens within the instrument, where fragment ions are generated. And then after the generation of the fragment ions, they're transferred to the mass analyzer, to the Orbitrap, where we can perform the mass measurement at high mass resolution. And in this approach, what we do is something called HCD, or higher energy collisional dissociation. There is a whole set of parameters that can be dynamically adjusted in this instrument. And these include parameters like the MS1 and MS2 accumulation times. So what they're determined by is usually we define a specified number of ions that we want to fragment in the HCD cell or that we want to analyze in the Orbitrap. And these are set as targets. And we then let the instrument essentially fill the HCD cell or eventually the Orbitrap with that specified number of ions. And usually that should be very fast. If there's good intensity coming in at the front end, then this may be a matter of milliseconds to fill an HCD cell or an Orbitrap. But these times are inherently dynamic, meaning if we set a certain target number of 250,000 ions, then the length it takes to achieve that target depends on the signal that's coming from the ionization source. And depending on the strength of that signal, the overall scan cycle of the instrument may vary. So just to try to illustrate that a little bit further. So what we're showing here is the duty cycle of the instrument now, where we have initially the MS1 scan, the survey scan, to essentially look for any precursor ions that we may wish to analyze and generate an MS1 or survey spectrum. And then once we've generated this survey spectrum, we will want to fragment and therefore determine the sequences of n number of fragment ions. In this case, we've set the method to analyze 10 different peptide peaks. And after scanning through each of these 10 precursor ions, the whole cycle starts again. So another survey spectrum is generated. From that survey spectrum, another 10 fragment ions are selected, are generated in that way. And it means that there's a number of settings in that kind of method. So the actual number of target ions that we wish to fragment after each survey scan is known as the top n for this experiment. That essentially the number of precursor ions to be selected for MSMS fragmentation. Also the number of ions that we're trying to fill the HCD cell and then the Orbitrap with, respectively. And all of these mean that there is essentially a distribution of possibilities in terms of how long the survey scan or each of the MSMS fragmentation events may take. And that leads to the following. So just here shown as an example is an LCMS chromatogram where we've separated a complex peptide mixture after digestion. And we see a whole variety of signals coming off the chromatography column. And at a given retention time, we generate an MS1 spectrum that's shown here in the top panel. And I've highlighted here a number of precursor ions that could now be selected for fragmentation. Then shown here is a precursor ion selection window in red, where we can essentially isolate. You can imagine this is zooming in on this peak and then fragment all of the peptides that are forming part of this peak. And that generates a fragmentation spectrum here shown below, or MS2 spectrum. And the mass shifts between these fragment ions, they should now correspond to the different B and Y ions that can be generated from this peptide. Of course, the mass spectrometer will scan through these very quickly and does this n times over. And in this example here, let's say it was a top 12 experiment. So we run through the generation of N1, 2, 3, 4, 5, 6, up to 12 fragment ions, and all of these are recorded. That leads to the following. It means that the actual scan cycle of the instrument is inherently variable. It depends on the signal strength, the strength of the signal that comes from the chromatography column at a given retention time. And this is shown here in data from a real data set that was visualized with an R script created by Alex Montoya at the MRC London Institute of Medical Sciences. And what we see here, that in the busy part of the gradient, we tend to have longer scan cycle times, between 1.5 and 2 seconds. And they can actually be summarized and plotted across the entire data set. And that is shown here. We can see that the average scan cycle appears to be somewhere around 1.7, 1.8 seconds, but there's both shorter and longer scan cycles depending on the strength of the signal of these MS1 precursors. And then in total, you can also summarize all of the MS2 accumulation times. So essentially ask the question, okay, how long did we take for all of the MSMS spectra for them to be generated? And there's some from presumably quite intense precursor ions where this was a very quick process where this took 10 milliseconds or less. But then we'll have a whole bunch of spectra that successively take longer. And then we see here that there's a very high number of spectra that took a whole 50 milliseconds to generate. So 50 milliseconds in this experiment is because the instrument setting here specified that the maximum time we allow for this process to occur is 50 milliseconds. So if the target number of ions has not been reached, we set a hard cutoff and then move on to the next MSMS spectrum to be generated. So these kind of instrument settings, they are part of the MS acquisition method, and this is just showing a real example of one of these instrument acquisition methods. Essentially, there's a number of things that then determine this whole process of precursor ion selection and data generation in data-dependent acquisition. This is the level of intensity of the precursor ions. We can set rules about certain charge states being excluded, such as typically in proteomics, we exclude singly charged ions because very often these are contaminants. Then we can perform dynamic exclusion, where we basically try to avoid selecting the same peptides over and over again. So if you have a very abundant peptide, the instrument might be tempted to just keep on going back to that same peak, to that same signal. And essentially here we have a setting where we say, once it has been fragmented, it needs to be now excluded from fragmentation for a certain length of time. And then further settings include the width of that precursor selection window, potentially an offset to that window as well, and level of collision energy to fragment these peptides. And all of these settings can be changed and adjusted. So this is an error here. I apologize. So I wanted to move on to data-independent acquisition strategies now, and just to maybe highlight a few of the limitations of the approach that we've just looked at. Limitations of label-free quantification using data-dependent acquisition, or DDA, are that this precursor ion selection is inherently a stochastic process. So what we mean by that is that there's rules that govern the selection of these precursor ions, but there's no guarantee that we will actually comprehensively be able to cover all of the precursor ions that have a potentially sufficient intensity due to limitations of scan speed of the instruments or the complexity of the sample. And there's certain methodologies that can be employed to improve on that. One of these is a methodology called match between runs, where essentially we say that if we've got a confident identification of a particular peptide signal in a given LCMS run, we can transfer that identification to a different LCMS run, as long as it has that particular MS1 signal has the same accurate mass and the same retention time as the signal in the previous run or another run within the same set. But that has to be restricted, so that should only be done, this matching between the runs, if it's really the same accurate mass within a very narrow window and the same retention time within a narrow window. But this allows us to transfer identifications across to essentially fill in some of the gaps that may be created by that particular peptide being present at a lower level in another sample in another LCMS run. And if we have further criteria that we can use, such as an ion mobility drift time, we can make this matching process even more confident by having more and more conditions that need to be matched and need to be allowed in order to enable this transfer of identifications across different runs. So what I would like to move on to now is a different approach that has been termed data-independent acquisition, or DIA. And what I'm showing here is a very early approach of that was utilized and was termed MS to the E acquisition, or high-low collision energy switching. And here the fundamental idea was to essentially not to perform any precursor ion selection, but to only cycle between two states where we have either a low collision energy and generation of MS1 data, or high collision energy and generation of MS2 data. but not actually separating out different precursors from which to generate this MS2 data. So this is shown here as an example where at the low collision energy state for these two molecules A1 and B1, we get one mass peak each that would be at a different intensity level and a different mass. And in this example, they also occur at a different retention time in the chromatogram. And then when we're in the high collision energy setting, what we generate is MS2 data. And if only A1 is eluting off the column, we will obtain a fragmentation spectrum of A1. And if only B2 molecule B2 is coming off the column, we only generate a fragmentation spectrum of B2. So in this fairly simplified setting, that works straightforward. But what happens if maybe these things elute closely to each other, or where there's partial overlap of the chromatographic profiles? And that is shown over here. So here we could have a situation where the two molecules are actually partially overlapping, and we still generate MS1 and MS2 level data with the respective MS1 spectra and MS2 spectra. But we now can actually assign which fragment ions belong to molecule A and which fragment ions belong to molecule B, not through the process of precursor ion selection as previously, but simply by observing the chromatographic behavior of the intensities of both the precursor ion and the respective fragment ions. And that is sort of plotted over here, where we say, okay, as long as they describe the same shape in terms of peak shape on the chromatography column, it means that the signal A1 should belong to all of the signals from A2, and B1 precursor ion signals should belong to the fragment ion signals of B2. So essentially what we're saying is we may be able to do away with selecting specific precursor ions. And that was introduced in this MS to the E methodology on Waters instruments. Now, of course, there is some limitations to this approach, which is if there is very high complexity, the fragmentation data that's generated might become rather complex and might really challenge this concept here to some extent. And to improve on that further, the following data independent acquisition strategy was introduced, and an early iteration of that approach was this swath MS, or sequential windowed acquisition of all theoretical fragment ion mass spectra was introduced. And there the idea was that rather than just having fragmentation of either having intact MS1 data or fragmentation of all of the precursor ions at a given retention time, we can divide up the M over Z range into equal sized fragments. And what is shown here is that we've divided this up into 25 different fragments. In this example, 25 different M over Z ranges. And what we're now going to do is we're going to perform selection of these windows that are now wider than the normal precursor ion selection windows in DDA, but which could still contain more than one precursor ion. And we have a slight overlap, in this case here, of 2 M over Z between each of the windows. So this is to avoid boundary effects where there's a peak that just happens to be between the two windows and therefore is not properly covered. And the idea now is that if we are able to scan quickly enough through all of these windows and perform the fragmentation, we may be able to generate data where we didn't need to perform precursor ion selection, but still have acquired MS2 data for all of the signals. And then an obvious question one could ask is, okay, is this going to be reasonable? Is it going to be a reasonable proposition to do this on a quick enough timescale to do this in the context of an LTMS run? And this may indeed be the case. So if each of these selection windows takes 50 milliseconds to acquire, and we have 25 of them, we would have essentially a duty cycle of 1.25 seconds plus the length it takes to generate the survey scan. And that would still be an acceptable scan cycle time. So this just as an introduction to the different acquisition approaches that can be used. And DRA is definitely one approach that's becoming more important and finds entry into practice in proteomics labs. So what I will spend time on now is to show the label-free quantification process in MaxQuant in a little bit more detail. So essentially, if we generate a data set from n number of samples and run each of these separately on the Tandem Mass Spectrometer, we generate a large data set of numerous sets of raw data. And for this whole data set, we now will perform feature detection. So basically identifying the MS1 peaks that are present, looking at all of the MS fragmentation spectra and doing a search. And there's this so-called first search that allows us to identify very confident identifications of peptides. And these highly confident identifications are now used to essentially perform recalibration of two things: recalibration of the retention time, but then also recalibration of the mass accuracy. And the idea here is that mass accuracy can drift over the length of a chromatographic gradient, and we can recalibrate any drifts in mass accuracy across the length of the chromatographic gradient, and that's what MaxQuant does. This is then followed by the main Andromeda search. So Andromeda is essentially the search algorithm incorporated into the MaxQuant software and is equivalent to the MASCOT search algorithm, but it was standalone, but it's a licensed software. Essentially now we're trying to match essentially sequences to as many of the MS fragmentation spectra as possible that were generated in this experiment. And then once these have been assigned, there's actually something called a second peptide search that allows us to find possibly co-isolated peptides, so where there were two precursor ions that were very close to each other, generating an overlapping MS spectrum which contains two sequences. And then at the end of the whole process, including any label-free quantification steps, we then generate a set of MaxQuant op tables, and these can then be analyzed further through statistical data analysis. And this is just showing that both Andromeda and MASCOT scores are highly correlated. So these two search algorithms perform rather similarly. And just to highlight that you know what the meaning is of this second peptide search that was mentioned on the last slide. So here's an example of two signals that were overlapping in the 2D chromatogram, and where co-isolation of these two precursors did indeed occur. That leads to a mixed MS spectrum which contains two peptide sequences. And the second peptide search essentially allows us to match another peptide sequence to this spectrum after the first set of fragment ions has been assigned and subtracted from that. And that allows us to increase the overall number of identifications from a complex sample. So, MaxQuant in 2014 introduced this label-free quantification algorithm. And the philosophy of that approach really was to say, if we're comparing complex samples, we very often will have a situation where there is a large number of unchanging proteins, and they will generate peptides that will have very similar levels of intensity. And then there will be a few proteins which are present at high or low intensity. And essentially the idea now is for the MaxLFQ process is to use the unchanging background proteome to allow us to perform a normalization of the different ones. So the normalization has the purpose of normalizing any technical differences in the mass spectrometer or differences in loading across different samples. That is shown here as an example. So this is from a real data set where we've got samples A and B that were analyzed in technical duplicate. And we can now plot all of the protein ratios of all of the proteins across samples A to B, and we get a curve that looks like this, where we've got the upregulated proteins up here and the downregulated proteins below here, and then in between a lot of proteins that change only to a much smaller degree. And essentially what the LFQ algorithm now does is it shifts that whole distribution, so leading to a constant ratio for this distribution of data in the middle, where there's relatively little change. And just to summarize this, so essentially in MaxQuant there is a whole suite of tools that allow us to identify and quantify proteins through different methods, both label-free, SILAC, and Isobaric labeling. This generates a whole set of output tables, and of these, the one that's most frequently looked at is the protein groups table that summarizes now both the identification and quantification information at the protein level. And then the other software to mention is Perseus, and Perseus allows us to perform the statistical data analysis and visualization of this type of data. And I'll just show you this very briefly here. So this is an example of a very simple experimental design of 3 well-typed cells in 3 biological replicates, and then 2 clones, clone 3 and clone 7, in equally 3 biological replicates. These were all analyzed in a label-free quantification workflow, and then LCM-SMS performed on a Q-exactive mass spectrometer, and then analyzed in this way. And then the data analysis was performed in MaxQuant, and a total of over 6,700 protein groups across all samples were identified and quantified. So this is what this looks like now on the Perseus software. So we've got lots of protein intensities across all of the different samples. And we're now able to generate in Perseus scatter plots. So here we look at log transformed intensities, and we see that for 2 technical replicates, they're highly correlated. And we can also show this correlation with this heat map here, where we show that a lot of the points are on this diagonal, showing very high correlation between technical replicates. And then we can do this for the entire data set as well. And then we can assign Pearson's correlation coefficients to show the actual correlation between either biological or technical replicates. Then finally, we can also visualize intensity histograms for each of these runs, where we show high intensity proteins at the upper end of this distribution here, and low intensity proteins at the lower intensity end. And then we see that overall there's a normal distribution of protein intensities. And then finally, we can also quantify the number of missing values. And as shown here, there is actually quite a significant percentage of missing values detected. So proteins with a 0 intensity in this data set. And that is partially due to the fact that this is generated with the DDA data-dependent acquisition approach. And then finally, we can also highlight this data on a volcano plot. And what a volcano plot allows us to do is to show both the fold regulation on the x-axis and then the significance on the y-axis. And just to show an example of what that means. So if we've got a protein that is has a high fold change and is highly significant, that implies that both there is a large fold change, but also when determining the p-value for the significance of this change, it is such that it is significant after multiple testing correction. Whereas here we show an example of another protein where there is no large fold change, and therefore it's not a significant change between the two, despite the fact that we've still got very tight error bars, whereas here at the bottom end we've got both larger error bars and no fold change for a protein that's shown there on a volcano plot. Now, I think I've come to the end of my lecture because I've run out of time. So what I would like to encourage you to do is to ask any questions in the Q&A window, and I will take your questions now. Yeah. So I'll just read out the first question that I've got here. So this is, which is the difference between CID and HCD fragmentations? Do these two fragmentations lead to the formation of different fragment ions from the same precursor peptides? So it happens that CID and HCD fragmentation are actually quite similar in the fragment ions they generate. So we still expect predominantly B and Y ions to be generated. The difference is that this HCD type fragmentation, standing for higher energy collisional dissociation, is also known as beam time CID, is essentially the implementation of a CID approach on an Orbitrap-based mass spectrometer. So essentially, a Q-exactive mass spectrometer, which has an HCD cell, essentially technically has to solve the problem slightly differently in how the fragmentation is induced. And therefore that has a different name, but the outcome is actually extremely similar between these two fragmentation approaches. However, there are other approaches that give you different fragment ions. So another example is electron-based fragmentation approaches like EGD, electron transfer dissociation, and there you generate C and Z ions. So you essentially break the polypeptide at a different place, at a different bond. Do we have any other questions? If not, then it remains for me to thank you for attending the lecture, and feel free to contact me if you have any questions at a later stage. And I always encourage anyone to approach us in an early stage in a project if you want to perform proteomics analysis with the MRC LMB proteomics facility. Thanks for your attention.